before I dive into the mechanics of how cells divide, I think it could be useful to talk a little bit about a lot of the vocabulary that surrounds DNA. There's a lot of words, and some of them kind of sound like each other, but they can be very confusing. So the first few I'd like to talk about is just about how DNA either generates more DNA, makes copies of itself, or how it essentially makes proteins. And we've talked about this in the DNA video. So let's say I have a little, I'm just going to draw a small section of DNA. Let's say I have an a G, a T. Let's say I have two T's, and then I have two C's. Just some small section. It keeps going. And of course, it's a double helix. It has its corresponding bases. Let me do that in this color. So A corresponds to T, G with C. It forms hydrogen bonds with C. T with A, T with A, C with G, C with G. And then, of course, it just keeps going on in that direction. So there's a couple of different processes that this DNA has to do. One is when you're just dealing with your body cells and you need to make more versions of, let's say, your skin cells, your DNA has to copy itself. And this process is called replication. You're replicating the DNA. So let me do replication. So how can this DNA copy itself? And this is one of the beautiful things about how DNA is structured. Replication. So I'm doing a gross oversimplification, but the idea is these two strands separate, and it doesn't happen on its own. It's facilitated by a bunch of proteins and enzymes, but I'll talk about that, the details of the microbiology in a future video. So let me make separate. So these guys separate from each other. Let me put it up here. They separate from each other, and let me take the other guy. Not too big. The guy looks something like that. They separate from each other. And then once they've separated each other, what could happen? Let me delete some of that stuff over here. Delete that stuff right there. So you have this double helix. They were all connected. They're base pairs. Now, they separate from each other. Now, once they separate, what can each of these do? They can now become the template for each other. So now, I can form a, if this guy's sitting by himself, now all of a sudden a thymine base might come and join right here. And so you could have these nucleotides will start lining up. So you'll have a thymine and a cytosine, and then an adenine, adenine, guanine, guanine, and it'll keep happening. And then on this other part that was formerly, this other green strand that was formerly attached to this blue strand, the same thing will happen. You have an adenine, a guanine, a thymine, thymine, cytosine, cytosine. So what just happened? By separating and then just attracting their complementary bases, we just duplicated this molecule. Right? And I'll go more, we'll do the microbiology of it in the future. But this is just to get the idea. This is how the DNA makes copies of itself. And especially when we talk about mitosis and meiosis, I might say, oh, this is the stage where the replication has occurred. Now, the other thing that you'll hear a lot, and I talked about this in the DNA video, is transcription. Transcription. In the DNA video, I didn't focus much on how does DNA duplicate itself. But one of the beautiful things about this double helix design is that it really is that easy to duplicate itself. You just split the two strips, the two helices, and then they essentially become a template for the other one. And then you have a duplicate. Now, transcription is what needs to occur for this DNA eventually to turn into proteins. But transcription is the intermediate step. It's the step where you go from DNA to mRNA. And then that mRNA leaves the nucleus of the cell and goes out to the ribosomes. And I'll talk about that in a second. So we could do the same thing. So this guy, once again, during transcription, will also split apart. So that was one split there. And then the other split is right there. But instead of, and maybe it makes more sense just to do one half of it. So let me delete that. So let's say that just the green, this is the coding. Let's say that we're just going to transcribe the green side right here. Let me erase all of this stuff, right? Nope, wrong color. Erase this stuff right here. Now, what happens is instead of instead of having deoxyribonucleic acid nucleotides pair up with this DNA strand, you have ribonucleic acid or RNA pair up with this. And I'll do RNA in magenta. And so the RNA will pair up with it. And so thymine on the DNA side will pair up with adenine, guanine. Now, when we talk about RNA, instead of thymine, we have uracil, uracil, cytosine, cytosine. And it just keeps going. This is mRNA. mRNA. Now, once this, and then this separates, that mRNA separates, and it leaves the nucleus. It leaves the nucleus. And then you have translation. Translation. That is going from the mRNA to, you remember in the DNA video, I had the little tRNA, the transfer RNA, kind of were the trucks that drove up the amino acids to the mRNA, and this all occurs inside these parts of the cell called the ribosome. But the translation is essentially going from the mRNA, RNA, to the proteins. And we saw how that happened. You have this guy. Let me make a copy here. This guy separates. Let me actually copy the whole thing. This guy separates, leaves the nucleus, and then I'll delete this stuff. And then you had those little tRNA trucks that essentially drive up. So, maybe you have some tRNA trucks. You see adenine, guanine, guanine. This is tRNA. That's a codon. A codon has 3 base pairs. And attached to it, it has some amino acid. And then you have some other piece of tRNA. Let's say it's a uracil, cytosine, adenine. And attached to that, it has a different amino acid. And this occurs in the ribosome. And then the amino acids attach to each other. And then they form this long chain of amino acids, which is a protein. So just to summarize, the proteins form these weird and complicated shapes. So just to kind of make sure you understand all. So if we start with DNA, and we're essentially making copies of DNA, this is replication. You're replicating the DNA. Replication. Now, if you're starting with DNA, and you are creating mRNA from the DNA template, this is transcription. Transcription. You're transcribing the information from one form to another. Transcription. Now, when the mRNA leaves the nucleus of the cell, and I've talked, well, let me just draw a cell just to hit the point home. If this is the whole cell, and we'll do the structure of a cell in the future. If that's the whole cell, the nucleus is the center. That's where all of the DNA is sitting in there, and all of the replication and the transcription occurs in here. But then the mRNA leaves the cell, and then inside the ribosomes, which we'll talk about more in the future, you have translation occur, and the proteins get formed. So mRNA to protein is translation. You're translating from the genetic code, so to speak, to the protein code. So this is translation. So these are just good words to make sure you get clear and use them make sure you're using the right word when you're talking about the different processes. Now, the other part of the vocabulary of DNA, which when I first learned it, I found tremendously confusing, are the words chromosome. I'll write them down here because you can already appreciate how confusing they are. Chromosome, chromatin, and chromatid. So a chromosome is, so we already talked about, you can have DNA, you can have a strand of DNA. It's a double helix. Maybe this strand, if I were to zoom in, it's actually 2 different helices. And of course, they have their base pairs joined up. I'll just draw some base pairs joined up like that. So I want to be clear. When I draw this little green line here, it's actually a double helix. Now, that double helix gets wrapped around proteins that are called histones. That double helix gets wrapped around proteins that are called histones. So let's say it gets wrapped like there, and then it gets wrapped around like that, and it gets wrapped around like that. And you have here these things called histones, which are these proteins. These proteins. Now this structure, when you talk about the DNA as it is wrapped, or when you talk about the DNA in combination with the proteins that kind of give it structure, and then these proteins are actually wrapped around more and more, and eventually, depending on what stage we are in the cell's life, you have different structures. But when you talk about the nucleic acid, which is the DNA, and you combine that with the proteins, you're talking about the chromatin. Chromatin. So this is DNA plus, you could view the structural proteins that give the DNA its shape. Structural proteins. And the idea, chromatin was first used because when people look at a cell, every time I've drawn these cell nucleuses so far, I've drawn these very well-defined, I'll use the word, so let's say this is a cell's nucleus. I've been drawing very well-defined structures here. So that's one, and then this could be another one. Maybe it's a shorter, and then it has its homologous chromosome. So I've been drawing these chromosomes, right? And each of these chromosomes, I did in the last video, are essentially these really long structures of DNA, long chains of DNA kind of wrapped tightly around each other. So when I drew it like that, if you zoomed in, you would see one strand, and it's really just wrapped around itself like this. And then it's homologous chromosome. And remember in the variation video, I talked about the homologous chromosome essentially codes for the same genes, but it has a different version. And it came from, if the blue came from the dad, the red came from the mom. But it's coding for essentially the same genes. So when we talk about this one chain, let's say this one chain that I got from my dad of DNA in this structure, we refer to that as a chromosome. Chromosome. Now, if we refer generally, and I want to be clear here, DNA only takes this shape at certain stages of its life when it's actually replicating itself. Not when it's replicating, when the cell is dividing, DNA, before the cell can divide, DNA takes this very well-defined shape. Most of the cell's life, when the DNA is actually doing its work, when it's actually creating proteins, or proteins are being essentially transcribed and translated from the DNA isn't all bundled up like this. Because if it was bundled up like this, it would be very hard for kind of the replication and the transcription machinery to get onto the DNA and make the proteins and do whatever else. Normally, DNA, let me draw that same nucleus. Normally, you can't even see it with a normal light microscope. It's so thin that the DNA strand is just completely separated around the cell. I mean, I'm drawing it here so you can draw it. Maybe the other one is like this. Right? And then you have that shorter strand that's like this. And so it's all, you can't even see it. It's not in this well-defined structure. This is the way it normally is. And then you have the other short strand that's like this. So you would just see this kind of big mess of a combination of DNA and proteins. And this is what people essentially refer to as chromatin. Chromatin. So the words, it can be very ambiguous and very confusing, but the general usage is when you're talking about the well-defined one chain of DNA in this kind of well-defined structure, that is a chromosome. Chromatin can either refer to kind of the structure of the chromosome, the combination of the DNA and the proteins that give the structure, or it can refer to this whole mess of multiple chromosomes and DNA, of which you have all this DNA for multiple chromosomes and all of the proteins all jumbled together. So I just want to make that clear. Now, the next word is, well, what is this chromatid thing? What is this chromatid thing? And actually, just in case I don't remember if I labeled these proteins that give structure to the chromatin, or that make up the chromatin, or give structure to the chromosome, they're called histones. Histones. And there are multiple types that give structure at different levels, and we'll do that in more detail. So what's a chromatid? So when DNA replicates, so let's say that this was my DNA before, right? When it's just in this normal state, I have one version from my dad, one version from my mom. Now let's say it replicates. So my version from my dad, at first it's like this. It's a big strand of DNA. It creates another version of itself that is identical if the machinery worked properly. And so that identical piece will look like this. And they actually are initially attached to each other. They're attached to each other at a point called the centromere. Centromere. Now, even though I have two strands here, they're now attached. And when I have these two strands that contain the exact, so I have this strand right here. And then I have, well, let me actually draw it a different way. I could draw it multiple different ways. I could say this is one strand here. And then I have another strand. I have another strand here. Now, I have two copies. They're coding for the exact same DNA. They are identical. I still call this a chromosome. I still call this a chromosome. Chromosome. This whole thing is still called a chromosome. But now each individual copy is called a chromatid. So that's one chromatid. Chromatid. And this is another chromatid. Sometimes they'll call them sister chromatids. Maybe they should call them twin chromatids because they have the same genetic information. Chromatid. So this chromosome has two chromatids. Now, before the replication occurred, or the DNA duplicated itself, you could say that this chromosome right here, this chromosome like our father, has one chromatid. You could call it a chromatid, although that tends to not be the convention. People start talking about chromatids once you have two of them in a chromosome. And we'll learn in mitosis and meiosis, these two chromatids separate. And once they separate, that same strand of DNA that you once called a chromatid, you now call them individually chromosomes. So that's one of them. And then you have another one that maybe gets separated in this direction. And let me make that, circle that one with the green. So this one might move away like that. And the one that I circled in the orange might move away like this. Now, once they separate and they're no longer connected by the centromere, now what we originally called as one chromosome with two chromatids, you will now refer to as two separate chromosomes. Or you could say, now you have two separate chromosomes, each made up of one chromatid. So hopefully that clears up a little bit some of this jargon around DNA. I always found it quite confusing. But it'll be a useful tool when we start going into mitosis and meiosis and I start saying, oh, the chromosomes become chromatids. And you're like, wait, how did one chromosome become two chromosomes? And, how did a chromatid become a chromosome? And it's all just revolves around the vocabulary. And I would have picked a different vocabulary than, calling this a chromosome and then calling each of these individually chromosomes. But that's the way we have decided to name them. And actually, just so in case you're curious, you're probably thinking, where does this word chromo come? I don't know if you're old Kodak film, it's called chromocolor. And chromo essentially relates to color. I think it comes from the Greek word actually for color. And it got that word because when people first started looking in the nucleus of cell, they would apply dye. And these things that we call chromosomes would take up the dye so that we could see it well with a light microscope. So that's why and some comes from soma for body. So you could kind of view it as colored body. So that's why they call it a chromosome. So chromatin also will take up, well, I won't go into all of that as well. But hopefully that clears a little bit this whole chromatid, chromosome, chromatin debate. And we're well equipped now to study mitosis and meiosis.